Hello everyone, myself Dr. Meagle S. Matthew. Today I am going to present the second lecture of nanotechnology. In this lecture, I am going to discuss about transmission electron microscopic technique. It is considered as one of the important electron microscopic technique used for the characterization of nanomaterials. The electron microscopic technique uses a beam of electrons to create the image of a microstructure of a material with much higher magnification and resolution, which is much higher than that of light microscope. The high resolution electron microscope uses short wavelength of electrons for microscope illumination. The wavelength of electrons in electron microscope is about 10,000 times shorter than that of visible light. Such high resolution makes electron microscope extremely useful for the revealing ultrafine details of material microstructure. There are two types of electron microscope: transmission electron microscope and scanning electron microscope. In this lecture, I mainly discuss about the transmission electron microscopic technique and its instrumentation techniques. Transmission electron microscopy is a microscopic technique in which a beam of electron is transmitted through an ultra-thin specimen and an image is formed from the interaction of the electrons transmitted through the specimen, and the image is then magnified and focused onto an imaging device such as fluorescence or on a layer of photographic film, or to be detected by a sensor such as CCD camera. The figure shows the photograph of TEM instrument and the images taken from the instrument. Coming to the instrumentation setup, it has the following components along its optical path: electron source, condenser lens, specimen stage, objective lens and projector lens. So we can discuss each part by one. Coming to the electron source, the electron gun are used to generate electron source. An electron gun generates a high energy electron beam for illumination, and in the electron gun, electrons are emitted from a cathode, a solid surface, are accelerated by high voltage to form a high energy electron beam. There are two basic types of electron gun. One is thermionic emission gun and field emission gun. In thermionic emission gun, tungsten filament gun and lanthanum hexaboride gun are used. And in tungsten filament gun, the tungsten filament is heated to approximately 2700 Kelvin, and lanthanum hexaboride gun, the rod is heated around 2000 Kelvin. Then electrons are boiled off from the tip of the filament. And in field emission gun, in this type of electron gun does not need to provide thermal energy for electrons to overcome the surface potential barrier of electrons. The electrons are pulled out by applying a very high electric field to a melted surface. A tunneling effect instead of a thermionic effect makes the metal electrons in the conducting band cross the surface barrier. All lenses in an electron microscope are electromagnetic. The TEM has three lenses: condenser lens, objective lens and intermediate lens. There are two condenser lenses to demagnify the electron beam emitted from the electron gun, and the condenser lens system controls the beam diameter and convergence angle of the beam incident on a specimen. During operation, illumination area of a specimen and illumination intensity can be adjusted by controlling the current in the electromagnetic lens of a condenser. The intermediate lens is used to switch the TEM between an image mode and a diffraction mode. For the image mode, the intermediate lens is focused on the image plane of the objective lens, and the diffraction mode is focused on the back focal plane. The objective lens controls the final focus of the electron beam by changing the magnetic field strength. The TEM specimen must be a thin foil because they should be able to transmit electrons. That is, they are electronically transparent. A thin specimen is mounted in a specimen holder in order to insert into the TEM column for observation. The smallest specimen can be mounted on a TEM grid, and the grid is usually made from copper. Also, a copper mesh can be coated with a thin film of amorphous carbon in order to hold the specimen even smaller than the mesh size. For a liquid sample, the sample can be drop casted into the TEM grid. Another fundamental requirement for TEM is the high vacuum is required. That is, if the column is in gas filled environment, the collision between high energy electrons and air molecules significantly absorb electron energy, and the electron will be scattered by gas molecule, which will leads to the reduction of the beam intensity and stability of the electron beam. Or other gas molecule which could come from the sample or microscope itself, it will form a compound and condense on the sample, and it will destroy the sample, and the image is not in perfect form. So these are all about transmission electron microscopic technique, and thank you for listening.